My name is Jens-Uwe Ulrich. I'm a PhD candidate at the Hasso-Plattner Institute in Potsdam, Germany. And in this video, I'd like to present you Taxor, which is our new tool for taxonomic classification of long reads. Microorganisms are everywhere. We know that the mixture of microbes or the microbiome on and in the human body plays an important role in human physiology and diseases. For studying microbiomes, metagenomic sequencing has become a key technology. And the workflow for this looks like the following. So we take a sample from the human gut and then we extract all the DNA in that sample and construct a sequencing library. Then we put that sample on the sequencing machine, and what we finally get is a bunch of sequencing reads for which we want to know from which species they originate. So we use software tools for this taxonomic profiling step that determine the origin of the reads, and if we finally get that, we can also estimate the abundance of the species in the sample. One technique used for taxonomic profiling is the so-called pseudomapping, where we first create all possible substrings of size k from the sequences, which are called k-mers. Using these sets of k-mers, we can calculate individual and joint set cardinalities, from which we can determine set similarity scores like the Jacquard index, or in our case the containment score. In our example, we want to know the number of joint k-mers between a read and the reference sequence, and simply divide it by the number of all k-mers from the read. And from that we can infer the biological relatedness of the read and the reference. To calculate set similarities, data structures for approximate membership queries have become very popular. In particular, modified versions of Bloom filters. However, one of the main problems in taxonomic profiling is the ever-increasing size of the reference databases, which call for data structures that use less memory while still providing fast querying. One of the approaches that try to reduce the size of the index database is using a more compact filter data structure like the XOR filter that was developed by Thomas Graf and Daniel Lemire. The XOR filter is a space-efficient alternative to other data structures for approximate membership queries. It reduces the space overhead by more than 20% in comparison to Bloom filters, as we can see here in the left figure. And this can be translated to saving more than 2 bits, here like the 9.8 bits per key, compared to the 12 bits here for the Bloom filter when both have the same false positive rate. Indexing a given reference sequence using an XOR filter works as follows. We first generate all possible k-mers of the sequence and initialize an array of 8-bit values with zeros. When we want to insert a k-mer into our XOR filter, in this case a 13-mer, we need to calculate first an 8-bit fingerprint value for that k-mer. Then we calculate 3 hash values for the k-mer using 3 different hash functions. Each hash value points us to a position in the XOR filter. Now we only need to set these 8-bit values of the 3 positions in such a way that the logical XOR of the 8-bit values equals the result of the fingerprint. The algorithm for building the index has to determine the order in which k-mers are inserted into the XOR filter first, starting with k-mers whose hash values point to positions not shared with other k-mers. Since we need one XOR filter per reference sequence, our final data structure will consist of several thousands of XOR filters when indexing, for example, the whole NCBI viral RefSeq. However, we want to avoid several thousand queries for each k-mer of a given sequencing read we want to taxonomically classify. Thus, we developed an interleaved XOR filter data structure that combines all single XOR filters in such a way that the entries of the XOR filters are ordered in an alternating manner. In this example here, we have 3 reference sequences, each indexed by one XOR filter. The resulting interleaved XOR filter starts with the first entries of the 3 XOR filters building the first subvector. This is followed by the second entry of each XOR filter building subvector 2. And we can then build this interleaved XOR filter in this manner. This compact data structure offers us the possibility to simultaneously query all XOR filters in one go. If, for example, we want to know all reference sequences that contain the 7-mer CAGGATT, we calculate 3 hash values using exactly the same hash functions as used for building the index. The values point us to subvectors in the interleaved XOR filter that hold 8-bit values for each reference sequence XOR filter. Then we calculate a logical XOR of the subvectors and the concatenated 8-bit value of the fingerprint. This results in an 8-bit value for each reference sequence, and if this value equals 0, the k-mer is contained in the respective reference sequence. And we set the appropriate bit representing the reference sequence in our binning bit vector. We repeat this query procedure for each k-mer of a given sequencing read and combine the binning bit vectors to a counting vector that stores the number of matching k-mers between the read and each reference sequence in the index. Unfortunately, interleaved filters have one main problem. The overall size of the data structure depends on the largest single filter, or in other words, for a set of reference sequences with highly diverged genome sizes, the space overhead can become significant. To overcome this issue, an hierarchical version of the interleaved Bloom filter data structure has recently been described, where large filters can be split into several smaller ones and small filters can be merged into one larger filter, which itself is another interleaved Bloom filter. We implemented the same approach to extend the interleaved XOR filter to an hierarchical interleaved XOR filter that works as follows. So building the HIXF requires to specify the maximum number of XOR filters on each level. So in our example, it's 5. Then we split the large reference genome A into 2 XOR filters. Genomes C and D as well as E to M are merged into one XOR filter on the highest level. Merged filters now represent individual XOR filters on the next level. For example, the merged genomes of C and D will be stored in an interleaved XOR filter that contains of 3 XOR filters on level 2. This whole process is recursively repeated until there are no more merged bins in an interleaved XOR filter in the leaves of this tree-like structure. The hierarchical structure not only saves a lot of space, it also reduces the query time. So let's pretend we have a long nanopore read, and dependent on the read length and the number of k-mers generated from that read, the threshold to consider this read a match with one of the reference genomes in our index database is set to 3241. We first query the level 1 interleaved XOR filter and see that only the XOR filter of the merged genomes E to M exceeds the minimum number of matching k-mers with that read. Then we go to the next level and query the interleaved XOR filter representing genomes E to M. Then we recognize that only the XOR filter for genomes H to J show the minimum number of k-mer matches with the read, and thus we have to query the appropriate filter on the third level, which finally results in a match between reference genome H and our given nanopore read. We implemented the described data structure into a new tool which we call Taxor. On this slide you can see a typical workflow for the use of Taxor. The first step is building an indexed database from a large set of reference genomes. And then we query a set of long reads against the indexed databases. If there are several sets of large databases, it can be helpful to create several indexed databases and query them separately. The search results of the query against the indexed databases then have to be merged in this scenario. And finally, a profiling step refines the taxonomic classification and reports binning and abundance estimations for the queried sample. For further reduction of index sizes, k-means selection schemes like minimizers have become popular over the last decade. But instead of minimizers, we decided to use sync-mears because recent studies have shown that sync-mear schemes improve mapping under higher error and mutation rates like we would expect with long nanopore reads. But the question is how many sync-mear matches one could expect for a correct read to reference match, given that the user can define different parameter values for the k-mear size and different error rates for the query read set. To overcome this issue, we empirically derived thresholds for the minimum number of sync-mear matches to consider a read as a match. Therefore, we randomly selected 1000 bacterial sequences from the GTDB database and created an HiXF index of the sequence set with different sync-mear sizes. Then we simulated nanopore reads with different read accuracy rates between 80 and 99%. Then we queried the reads against all indexes and determined the minimum fraction of matching sync-mears for each combination of sync-mear size and read accuracy, which we then stored as an internal matrix in Taxor. So let's pretend we queried a set of sequenced nanopore reads against our index database, and we already obtained search results. The first thing we need to do is refining our search results by filtering out spurious matches, in particular where we have reads that have matches to many different references. For this purpose, I implemented a 3-step filtering approach that first gets rid of all read to reference matches where the containment score, or the ratio of matched sync-mears to all sync-mears of that read, is below 80% of the maximum containment score this read has with any reference in the database. Next, we will only consider reference genomes as part of the sample that have at least one uniquely matching read. Matches to other reference genomes are immediately filtered out. And finally, we applied a 2-stage taxonomy assignment algorithm from a tool called Megapath that merges reference genomes that share a certain amount of read-to-reference matches. In the last step of the profiling, a standard expectation maximization algorithm refines the read-to-reference matches and estimates sequence and taxonomic abundances for a given sample. To evaluate Taxor, we created a custom database of 21,003 species representatives from the NCBI RefSeq, consisting of 11,579 viral, 8,938 bacteria, 403 archaea, and 83 fungi genomes. Then we indexed this database using Taxor and 5 common taxonomic profilers using similar parameters for k-mear size, false positive rate, and the k-mear selection scheme. For investigating read classification metrics, we chose 2 publicly available nanopore real mock community datasets and 1 PacBio hi-fi mock community. For these datasets, we created a ground truth by mapping the reads to the reference genomes provided by the manufacturer of the mock communities. Now we calculated for each of the 3 real mock communities the read classification precision, recall, and F-scores using Taxor, Centrifuge, Gannon, KMCP, Kraken2, and MetaMaps. On this slide, we present the results on species level, and we can see that Taxor has the highest precision across all tools for the 2 nanopore datasets, while only KMCP is better for the PacBio dataset. However, KMCP has by far the worst recall of all tools. Regarding recall, MetaMaps performs best for the 2 ONT datasets, followed by Taxor and Gannon, while for the PacBio dataset, Taxor and Gannon are better than MetaMaps. Consistently, we see that Taxor is on par with MetaMaps with regards to F1 and F0.5 score. Although Taxor shows very good results for taxonomic classification of long reads, on species level our main intention was to reduce the computational requirements of taxonomic profiling and in particular the memory requirements. Thus, we measured time and memory usage for building the database index and querying a set of 426,000 nanopore reads for all 6 tools on a high-performance computing cluster. As we can see, Taxor is as fast as Kraken 2 when building the index and needs only half the memory of Kraken 2. Only KMCP outperforms Taxor regarding the computational requirements for index building. However, KMCP needs almost 37 minutes for querying the reads, while Taxor is as fast as Gannon and Kraken 2. Finally, Taxor shows by far the lowest memory requirements when querying the read set against the build database index, reducing the memory footprint by a factor of 2 to 3 when compared to the most popular tools Centrifuge and Kraken 2. Overall, Taxor shows the best combination of time and memory usage for index building and read querying. We can conclude that Taxor slightly improves the long-read taxonomic classification, and it is comparably fast as Kraken 2 and Gannon when querying long reads. It reduces the memory and the disk usage by more than 50%, and it could be a good alternative for real-time long-read taxonomic profiling. Taxor is available on publicly available on GitHub. And if you have questions, you can always write me an email. With that, I want to finish this short presentation and want to thank all the members of the Renard Lab, especially my advisor Bernard Renard and Jakub Bartoszewicz. And I also want to thank the members of the Sekan group at the Free University of Berlin, in particular Knut Reinhardt, Vitor Piro, Svenja Mirika and Enrico Seiler.